Hi, everyone. Thank you all for joining me today. And thank you to Simon and Eric for introducing me. Today, I'll be presenting my most recent work, which is on LIGAR-1 and LIGAR-2, two IclR type transcriptional regulators and how they control carbon compound utilization in Pseudomonas putida. So Pseudomonas putida is a gram-negative soil bacteria, and it's often associated with the plant rhizosphere, which is this narrow zone that surrounds the plant root. This region is rich in root exudate compounds, which accounts to roughly 20 to 40% of the plant's fixed carbon. These exudates include compounds like organic acids, sugars, amino acids, aromatic compounds, and flavonoids, and they help to shape and sustain the microbial community in this ecosystem. So in the rhizosphere, Pseudomonas putida forms a symbiotic relationship with the plant, and it supports things like nutrient uptake to help the plant require nutrients that has challenges. It assists with lateral root production through hormone production, and it also helps fight against phytopathogens. However, organisms in this ecosystem must compete for nutrients and space, making it a highly competitive environment. So carbon utilization is the cornerstone of microbial survival and proliferation, especially in a dynamic environment like the rhizosphere. So this region is highly chemically complex with nutrient availability constantly shifting. And to thrive here, bacteria must be able to adapt and detect changes in available carbon source and rapidly adjust their metabolic strategy. So this metabolic flexibility is essential, not just for them to be able to get energy, but to also maintain a competitive edge in the rhizosphere where they outcompete any competitors. So bacteria have evolved multiple strategies to optimize their carbon utilization. So in the first scenario, you have simultaneous utilization, in which case compounds like glucose and aromatics, they're utilized at the same time because the metabolic pathways that are present are active, they're both active. And this leads to a steady growth curve. In the second scenario, you have hierarchical utilization. And so here, bacteria prioritize one carbon source, in this case, the one that offers the highest energy yield, and suppress the metabolism of the second carbon source. And it's not until the first carbon source is depleted where you get the utilization of the second compound, but there is a temporary lag phase because the bacteria need to reprogram their metabolic pathways to enable utilization of the second carbon source. And then you have sequential utilization. In this case, you're still utilizing one carbon compound first and then the second, but the bacteria have already have the programmed pathways for the second one active, so they can just start immediately using it and there's no lag phase observed. So in this case, there are trade-offs to each. So simultaneous utilization offers speed, but hierarchical and sequential utilization allows bacteria to maximize energy efficiency. So here are some molecular mechanisms for supporting hierarchical and sequential utilization. So in Pseudomonas putida, you have the carbon catabolite repression system. So the proteins CRC and HFQ bind to distinct sequences on mRNA, and it prevents ribosome binding to inhibit translation for the genes that you do not want expressed for a specific pathway. When the first carbon source is depleted and you want to use the second carbon source, these two proteins get sequestered by CRCZ sRNA to enable translation. In E. coli, there's also something called the inducer exclusion method, which involves the phosphotransferase system. In this case, when you have both glucose and lactose, the E. coli preferentially use glucose, and the protein E2A-GLC will bind to the lactose promoter and inhibit it to prevent import of lactose. And then finally, you have transcriptional regulation. In this case, Bacteria activate or repress gene expression for their specific metabolic pathways. However, there's limited research on how transcriptional regulation operates when two carbon sources are controlled within a single transcriptional unit. So that gap in knowledge is the focus of today's presentation. We'll explore how bacteria coordinate dual carbon sources regulated at the transcriptional level. So aromatic carbon compound utilization and biosynthesis are the cornerstone of the Christian lab research. Through our investigation of shikimate pathway homologs, we identified the Lig1 and Lig2 operons. So the Lig1 operon has a shikimate dehydrogenase homolog, and shikimate dehydrogenase generally functions in the shikimate pathway by catalyzing the fourth step to make charismate, which is used in bacteria for aromatic amino acid biosynthesis. In the Lig1 operon, you also have two FAD binding oxidoreductases, a NIP-SNAP family protein, as well as an MFS transporter, which is annotated as a putative sugar transporter. And the Lig1 operon also has a divergently encoded Lig1 transcriptional regulator. Now, Lig2 operon is located about 150 base pairs from Lig1, and it contains a putative dehydroshikimate dehydratase, as well as a 4-carboxy 2-hydroxymucinate 6-semialdehyde dehydrogenase, or Lig-C. These two proteins are involved in the formation of protocatechate, and this Lig-C protein is one of the enzymes that's involved in the meta-cleavage of protocatechate into the final intermediates oxaloacetate and pyruvate for entry into the TCA cycle. Now, this Lig2 operon has a LIGR2 regulator, and given the proximity of these two operons, we hypothesize that they function together to regulate the import of sugar and aromatic compounds, as well as the metabolism of aromatics. So to investigate how LIGR1 and LIGR2 regulate these carbon compound import and utilization, we performed electromobility shift assays to test their ability to bind to their own promoter as well as each other's promoter. And what we observed is that the LIGR1 transcription regulator can bind to the Lig1 promoter, but cannot bind to the Lig2 promoter, whereas LIGR2 can bind to both Lig2 promoter and Lig1 promoter, but this interaction does not reach saturation even at 1 micromolar concentrations. So next, we solved the crystal structure of LIGR1 at a resolution of 2.11 angstrom, revealing 4 molecules in the asymmetric unit. And this is consistent with a tetramer. Now, the structure follows a typical IclR transcription factor architecture, which is an N-terminal DNA binding domain, which has a helix-turn-helix motif. You have an effector binding domain at the C-terminal end, and these two domains are connected by this alpha helical linker. Now, within the effector binding domain, we observed density for an acetate molecule, likely from the crystallization conditions that was embedded within the 7-stranded antiparallel beta sheet. And because we had the tetramer structure, we were able to see where these proteins dimerize and tetramerize, and we can see that this mostly occurs within their DNA binding domain. And finally, we compared the effector binding domain architecture between the crystal structure of LIGR1 and the AlphaFold model of LIGR2. And there shows strong overlap in terms of their 3-dimensional structure, but there are some minor variations. For instance, the LIGR2 regulator has this additional alpha-9 helix. So we investigated the phylogenetic distribution of LIGR1 and LIGR2 alongside well-characterized IclR transcription regulators, and our goal was to determine whether they cluster with known regulators to suggest potentially any functional similarities. And we found that LIGR1 clusters closely but independently with POBR, which is an IclR from Acinetobacter, which binds to 4-hydroxybenzoate for its utilization. And in contrast, LIGAR2 did not cluster with any of the known IcLRs, suggesting that it has a distinct role that has not yet been identified. And when we compare the binding site between LIGAR1 and LIGAR2, they share very minimal sequence conservation. In this case, the only residues that do tend to overlap is the ones that I've highlighted here in the stars. So the ones that coordinate this carboxylate of acetate, as well as proline. So these differences may underline their divergent regulatory roles. So to determine the regulatory function of LIGAR1, we conducted reporter studies using a beta-galactosidase reporter assay. And in this case, the expression of beta-galactosidase is directly proportional to the activity of the promoter region you have it fused to. And in this case, we monitor the production of orthonitrophenol, which produces this yellow color that we can then monitor via absorbance and then plot using Miller units, which averages the absorbance of the yellow color by the optical density of the bacteria. So these are simplified versions of the beta-gal experiments that I conducted from the original manuscript, but I'll just go through a brief summary of like the major finding. So under wild-type conditions, the expression of the LIG1 operon increases when 4-HBA is added as an inducer. And when LIGAR1 is either overexpressed or knocked out, we get a reduction in the expression of the LIG1 operon. In contrast, when we complemented the gene back into the bacteria, the expression of the LIG1 operon increased. And similarly, when we overexpress LIGAR2, we see that the expression of the LIG1 operon increases in the presence of 4-HBA. Now, because IcLR transcriptional regulators have an autoregulation mechanism to influence their own expression, we also looked at that. And in the case of the LIGAR1 regulator, we can see that the LIGAR1 regulator has high expression under glucose, and this is further induced when you add 4-HBA. And similarly, when we knocked out LIGAR1, the expression of the LIGAR1 regulator, it decreases, but it's not completely suppressed. So these overall indicate that LIGAR1 and LIGAR2 activate expression of the LIG1 operon. So we conducted the same experiment for the LIG2 operon and showed that 4-HBA is a potent inducer of the LIG2 operon. And this actually is also the case for quinate and protocatechate, but I'm only showing 4-HBA in the case of simplicity. And in this case, when you overexpress either LIGAR1 or LIGAR2, it suppresses the expression of the LIG2 operon. In contrast, when you knock out LIGAR2, the expression of the LIG2 operon is more potently induced. And when you complement that back, you get complete inhibition of the expression of the LIG2 operon, indicating that LIGAR2 is a repressor. And interestingly, we also observed that for the LIGAR1 expression, when you knock out LIGAR2, the expression of the LIGAR1 regulator is suppressed, but when we complement it back, it potently activates it. So not only does LIGAR2 repress the LIG2 operon, it actually also activates LIGAR1 expression. So next, we decided to look at the effects of 4-HBA on the biochemical properties of LIGAR1. And we did this using differential scanning fluorimetry as well as EMSAs. So DSF, we monitor the stability of the protein through its melting temperature. And when we added 4-hydroxybenzoic acid, we see an increase in the melting temperature of the protein, indicating that it's more stable. And when we did EMSA analysis, we were actually able to increase the percentage of fraction-bound DNA from 50% to 80% when we added increasing concentrations of 4-HBA. Now, when we look at the binding site of LIGAR1 with POBR from Acinetobacter, which in this case is crystallized with 3-hydroxybenzoic acid, here we had docked 4-HBA. We modeled it in using the ligand fit program in Phoenix Suites that was used to identify the crystal structure. We can see that there's several overlapping residues, but one of the key residues that was determined within the POBR paper was this glutamic acid at position 217, which is thought to form a hydrogen bond with the OH group of 4-HBA. And in this case, the crystal structure has not undergone the appropriate conformation, so it is a little bit further away than you would expect when if it was crystallized with this ligand. We conducted similar experiments for IclR-2, and we showed that protocatechuate and 4-HBA have a destabilizing effect on the melting temperature of the protein. And interestingly, when we conducted EMSAs, normally under the wild type condition, you form these two bands in the EMSA. So one corresponds to the dimer and the other to the tetramer. However, when you add quinate, proto, or 4-HBA, you no longer see this tetrameric sized band, which indicates that these compounds have a destabilizing role for the regulator and might help remove it off the DNA so that it can facilitate expression. As we said before, LIGAR2 is a repressor. So since we do not have the crystal structure of LIGAR2, we did do some simulated binding of these ligands in the binding site using autodoc analysis. And this predicts an affinity value for the favorability of the binding. And among these three compounds, we see that the residues of arginine 99 as well as serine 196 are sort of common throughout each of these ligands. So next, we looked at the effect of LIGAR1 on the growth of the bacteria on sugar as well as aromatics, and I'll just go through these one by one. So when we knocked out LIGAR1, we observed that the bacteria have a severely cannot grow on glucose, indicated by this green line here. So we monitored the growth of the bacteria over a course of 24 hours, and we're just showing the optical density. And the graph on the right here is just an area under the curve measurement to have a more easier representation of. Okay, when we complemented the LIGAR1 gene back into the knockout, we were able to restore growth on glucose when we induced expression with IPTG. In contrast, the growth of LIGAR1 knockout and 4-HBA is reduced, but it's not completely inhibited. And when we tried to complement this phenotype by introducing LIGAR1 back, we were surprised to find that overexpression of LIGAR1 completely suppressed the growth of this bacteria on 4-HBA, indicated by this orange line here. And this was really difficult for me to understand at first because normally when we do these complementation studies, we would expect the restoration of the phenotype back to wild type. But in this case, no matter what I tried, it was always suppressing its growth. So I conducted a screen with the other compounds, and what we were able to show is that when you overexpress either LIGAR1 or LIGAR2 in the wild type background, it suppresses the growth of the bacteria on aromatics like 4-HBA, quinate, and proto. So indicating that the phenotype I was originally seeing in the knockout background when I complemented it was actually true. Now, when we look at the utilization of glucose and aromatics in the LIGAR2 knockout, we see a minor reduction in the ability for the knockout to grow on glucose, and same with the aromatics. But when we, again, overexpressed it, you see this similar phenotype of suppression. So to summarize the growth data, we have two scenarios. In the first case, you have the overexpression of either LIGAR1 and LIGAR2. And what we think is happening is that when you overexpress either, you get activation of the LIG1 operon, and this can facilitate sugar and aromatic amino acid or aromatic compound import, leading to normal growth on glucose. However, the LIG2 operon in this case is suppressed, and this prevents the utilization of the aromatics, and that's why you're seeing the suppression of growth on 4-HBA, quinate, and proto. In the case of the LIGAR1 and LIGAR2 knockout, in this case, the LIG1 operon is not able to be expressed as well. In this case, you have lower sugar import as well as aromatics. And then the Lig2 operon is no longer repressed. And so if there's any of these compounds that do get imported, they're able to be utilized by this second operon. Now, while I was conducting these growth assays, I did notice that the bacteria have, that they were morphologically different, because when I was growing them in the flasks, they would produce these like amorphous structures where they would clump together. So I decided to investigate that further. And I grew the cultures under static conditions, and I stained the bacteria with crystal violet and imaged them using confocal microscopy. And what we were surprised to see was that under the utilization of glucose and 4-HBA, the LIGR1 knockout has elongated phenotype, and they also form these aggregations. And the reason why we think that this may be happening is because these bacteria have a reduced ability to grow on these carbon sources. They may be forming these biofilms in order to sort of function as a protective mechanism away from the extracellular environment. And similarly, we conducted some studies to show that these bacteria undergo more cell lysis. So a lot of their protein content ends up in the extracellular space, maybe to provide some additional nutrients to use the compound that they cannot use in this environment. So just to summarize, the LIGR1 and LIGR2 regulators, they regulate the Lig1 and Lig2 operon. And what we think is happening is that in the rhizosphere, where you have, in a situation where you have low aromatics or high glucose, the Lig1 operon is expressed to enable import of both. However, most of this glucose is diverted to the ED pathway for glycolysis, or for energy production, sorry. And then any aromatic compounds that are imported, they get redirected to the shikimate pathway for production of charismate and the 3 aromatic amino acids. And this sustains normal growth. In this case, the Lig2 operon is repressed, and that's why you're seeing this redirection back to the shikimate pathway. However, if the environment changes and now you have high aromatics and low glucose, the energy that you get from the glucose pathway is no longer as prominent. And most of the aromatic compounds are now diverted away from the shikimate pathway, and expression of the genes from the Lig2 operon would facilitate breakdown of these compounds, which could then feed into the TCA cycle for energy. So the bacteria are constantly modulating the expression of this operon to either use aromatics or switch to glucose. And LIGR1 and LIGR2, like I said, are important regulators of carbon compound import and utilization, and it could be important for overall establishing dominance in the rhizosphere, as well as suppressing growth of competitors, especially if they do not have this operon. And we're currently looking at using the LIGR1 regulator, either the overexpression of this protein or the knockout. In terms of the valorization of carbon material, maybe lignin for industrially relevant compounds. In this case, if you want, for example, only sugar molecules, you could use the LIGR1 knockout. Or if you're more interested in the aromatics, you could use the overexpression where you're suppressing the utilization of those aromatic compounds. So I'd just like to acknowledge my lab for their support over the years to get to this point of finishing off this work, especially my professor, Dinesh Krishnadat over here, and some of my current and past lab members. Thank you all to listening to my presentation. And if you'd like to connect with me, you can use this QR code to find a link to my email and LinkedIn. And because I'm wrapping up my PhD studies, I'm open to either postdoc or industry opportunities in the new year as I wrap up at my lab here at U of T. Thank you so much.